Thank you so much for this opportunity to share what I've been doing for the last few years. And I'm going to be talking about lutein, and the title is From the Eye to the Brain. And the reason for that is most of what we do know about any kind of benefits that lutein has to do with the eye. And I'll be talking about some of the emerging data that we're accumulating on what it may be doing in the brain. So first of all, what is lutein? Just to bring us all on the same page, lutein is a carotenoid, and we find them throughout nature. We find them in plants, in our algae, and some of the fish because they eat the plants and algae. And they really are important, in nature. They really do serve a function, and I'll be talking about why I think lutein's important. So first of all, carotenoids in plants are really vital. And what they do is you got your visible light, you got your light spectrum, and light's damaging. We know that light causes damage, and these carotenoids absorb the light in the middle of that spectrum, wavelengths that chlorophyll doesn't take care of, such that it absorbs that light in that plant cell so it can't result in damage from that exposure to light. So that's really important. And so we're beginning to think that lutein is particularly important, especially in early life, and I'll talk a little bit about that. And I like this slide because it's an egg, right? And that's early life. And that egg is yellow. And so now everyone in this room should know why that egg is yellow, because it's lutein. That's lutein there. That's lutein in early life. So as I said at the beginning, lutein has a role in the eye, and I'm not sure if I can get this pointer to, anyway, so there's, in the back of your retina, so I'm looking at everyone right now through my macula. That's what your central vision is. So when you think about it, your retina, your macula that's in the center of your retina, that you have central vision, that's the part of your retina that's going to be particularly vulnerable to light damage. So your eyeball's not like your liver or your bones because it's vulnerable to light damage. So here you have a part of your retina, your macula, centrally located, particularly vulnerable to light damage because it's a direct hit. So when you think of ages, decades of light exposure to that retina, that's the part of the retina that falls apart. So you get age-related macular degeneration. Major cause of visual impairment in older adults. That with cataracts, but cataracts you just get a new lens. With the macula, once you have AMD, there's no cure. There's no treatment for that. So the idea is, how do you delay the progression? And that's where lutein comes in. Because I told you about the macula, vulnerable to light damage, and guess where you find lutein? You find lutein accumulates in the macula to the amount of 500 times more than anywhere in the body. And so that's great because lutein is an antioxidant. Light is oxidatively damaging. But it's also a blue light filter, and blue light, that's visible light. So if you have this yellow pigment, lutein, filtering that blue light, it protects those underlying photoreceptor cells, and there's a lot of good evidence to support that. So that's what we know about lutein, and there's been studies, the most recent being the AREDS-2 study that found that people with low lutein intake delay the progression of AMD if they're given more lutein. So that's pretty good. So here it is. That's your macula in the top slide, and you see the yellow? That's lutein. And it's isomer zeaxanthin. They're highly concentrated there. And the second slide shows you that it indeed does absorb that blue light. So it does provide the protection from light damage to your photoreceptor cells. So what about early life? What do we know about lutein in early life? Well, not a lot, actually, but there's a lot of reason to believe that it is going to be important. First of all, macular pigment levels, so macular pigment, lutein, zeaxanthin in the macula, that's what macular pigment levels, low levels may influence early visual development. And we've done studies with monkeys, and the reason we had to do studies with monkeys is because unlike your white rat, your gerbil, They don't have a macula. You really have to work with primates when you want to study lutein and its role in the eye. So when we had access to monkeys that had never seen a carotenoid in their life, even their mother has never seen a carotenoid. And when these monkeys, without carotenoids in their diet, you actually see an impairment of retinal pigment epithelial cells. And what those cells do, they're in the retina, they traffic nutrition going in and waste products going out. So in the absence of lutein and zeaxanthin, that distribution of those cells is impaired, and that's not good. The good thing was, though, when these monkeys were given lutein back, you saw repairment of that distribution. The other thing is that without macular pigment, you can expose that retina to damage from this blue light and oxidative stress, and that's pretty important. So that's all reviewed by Randy Hammond in Nutrition Reviews. So that's about light, about visual function, but what about cognitive function? So we really don't know a lot about that, but this is what we did, is we first wanted to find out, is lutein even there? Do you even find lutein in the infant brain? So we analyzed brains that we obtained from a brain bank from children that had died of sudden infant death, so they were otherwise healthy, and analyzed them for the dietary carotenoids. And so those are lutein, zeaxanthin, cryptoxanthin, beta-carotene, alpha-carotene, and lycopene. And when we looked at what was there in terms of carotenoids, if you look at those orange lines, that's lutein. So lutein was by far the major carotenoid in these infant brains. There was no alpha-carotene, there was no lycopene, and we analyzed 100 brains, there are only 2 that had lycopene in them. So lutein explained about 60% of the total carotenoids. So one way this could be true is that they're just eating more lutein. These children are eating more lutein. So we didn't have access to what they were eating, but we did have access to NHANES data to see what children in the United States are eating in that first year of life. So the pie chart on the right are the total dietary carotenoids, of which 12% is lutein. So looking at our data in brains, you see that it's almost 60% is lutein, indicating that there seems to be a preferential uptake of lutein into the brain, the infant brain. Very similar to what we see in the retina. Because I told you macular pigment, lutein, zeaxanthin, those are the only 2 carotenoids in the macula. We have hundreds in nature, about 30 in the diet, about 20 in circulation, but only lutein and zeaxanthin are in the macula, preferentially taken up into the macula, seeing the same kind of thing going on in the brain. So when we look at these data in infants comparing to older adults, we see that lutein is even a bigger chunk of the pie. It's suggesting that, if it does have a role in cognition, it may have a role in neural development as well. So what do we conclude from these studies in infant brains is that, 1, lutein is the predominant carotenoid in infant brain, accounting for more than half of the total carotenoids, suggesting that maybe it is important in neural development. So usually infant formulas are not routinely supplemented with lutein. It's not essential. If you don't eat it, you're not going to die. But it really does suggest that maybe there's should be further investigation into its impact on neural development. So why that? And so now I'm going to be talking about what we know as far as lutein's relationship in cognition. And that work is done in adults. And the first study I'll describe is macular pigment density, and it's related to cognition. So if you were paying attention, you'll know what macular pigment is. It's lutein embedded in the retina, right? It's lutein embedded in neural tissue. So we looked at that relationship in the Health, Aging, and Body Composition study, which is an NIH-funded study, in which we had 100 subjects who were recruited to the study because they were healthy and they were older. And there were 2 ancillary studies which there was macular pigment data collected as well as cognitive function. And when we looked at cross-sectional relationships with various measures of cognition for executive functioning, long-term memory, short-term memory, and so on, we found that macular pigment was significantly correlated with a lot of these measures of cognition. Not so much for serum levels of lutein and zeaxanthin. And serum levels is usually an indicator of short-term intake of these carotenoids, whereas macular pigment is a nice measure of long-term intake. So once it gets into the macula, it sticks around for a while. And so we have this measure of long-term lutein status, that is lutein in the macula, being significantly correlated with cognition in these older adults. So why does that make sense? Why does it make sense that if you have more in your retina, you're going to be smarter? And so we looked at that. We said, is this really a biomarker? And so that's a word we heard in the talk before us. Is it really a marker of what's in the brain? Because when you think about it, to get lutein from your food, to absorb it through the gut, have it circulate, and then go past that blood-brain barrier is kind of a trick. It has a lot of steps to take. So if it's going to get to the retina, it makes sense that it's going to get to the brain. So that's what we did. We measured matched retina tissue with brain tissue and looked at that relationship. And these are human data. And actually, this paper was just accepted, where you look at macular carotenoids, it's lutein and zeaxanthin, with the amounts in matched brain. So we looked at the occipital cortex of the brain, and it's just a gorgeous correlation. And there's not a lot of samples here, but we get a really high correlation, really suggesting that if I measure your macular pigment, which I can do in about 5 minutes without pinching or poking, and get your level macular pigment and have a really good indicator of what's in your brain. And that's terrific. That's really terrific, because then we can look at lutein interventions and see, you know, how it's affecting cognition and if we're getting it into the brain or the retina, because people do it better than others. And so that's a really nice thing to be able to have that kind of information. So the next study I'm going to describe is when we actually looked at brain levels of lutein and looked at its relationship with cognition in elderly. And this was part of the Georgia centenarian study, where we had subjects who, you know, upon, you know, giving consent for the study, agreed to donate their brains upon death. And so we looked at those brains, and we looked at pre-mortem measures of cognition, so measures that were taken within a year of death. We also had access to serum from a larger population of 300 adults, and we looked at relationships with cognition. So before I go into that, I like this slide a lot, because I talked about preferential uptake of lutein into the retina. And so this slide shows that happening in the brain. So on the left side, you see what's going on in serum, and those are your major dietary carotenoids. And so you have lutein, zeaxanthin, cryptoxanthin, beta-carotene, and lycopene. And these people, they were eating a lot of beta-carotene. So they were eating a lot of carrots and winter squash, not so much spinach. But when you look at in matched brain samples, lutein wins. Lutein is the major carotenoid, despite not being the major dietary carotenoid. Again, showing this preferential uptake of lutein into neural tissue. And this helps explain that. So, you know, this preferential uptake of lutein in the retina is explained by a binding protein there, and Paul Bernstein out of Utah has spent decades looking for this binding protein and found it in the retina. And whenever you have a specific binding protein for something, that's pretty important and gives a lot of biological plausibility for it being important. So I shipped off my brain tissues to Paul to analyze for this binding protein. And we looked at its relationship with lutein in these tissues. And what you see is there a really strong relationship in the pediatric brain tissue, very significant. Less so for the geriatric tissue, which isn't too surprising, because as we get older, there is more biological variability. But in any case, it really lends support for lutein being important, and it helps us understand why we're seeing this preferential accumulation in the brain. So then we went on to look at these brain tissues in the relationship with premortem measures of cognitive function, of which we did many measures. We also had a global deterioration scale, which is a measure of dementia. And the reason I mention this is that we were really interested in stages 1 through 3 of normally cognitively intact, some memory loss, some mild cognitive impairment. Stages 4 through 7 are all scales of dementia, and we weren't as much interested in those categories because once you get dementia, there's not a lot that nutrition can do. The real target population is earlier. That's where nutrition can have an impact. So that's what we looked at. So looking at the serum data, we looked at all the carotenoids, and we also looked at alpha tocopherol. Everything in yellow is significant. So lutein and zeaxanthin did pretty well. So people that were eating more of their green leafies had better relationships to cognition, whereas beta-carotene and alpha-tocopherol didn't do a lot. And I like to mention this is because both alpha-tocopherol and beta-carotene there have been clinical studies looking at interventions in relationship to cognition, and they really didn't show a lot, like these data. The other reason I like to mention alpha-tocopherol is because it's a major dietary antioxidant. And in our diet, there's much more alpha-tocopherol, vitamin E, than there is lutein. But you don't see that relationship there. So the idea being that if lutein's doing something with cognition, it might not be as an antioxidant. It might be doing something else. So these are the data we had in brain tissue. And again, it was lutein that had the significant relationship. So those that were normally cognitively intact had the highest levels of lutein in their brain. And we didn't see that for any other carotenoid, and we didn't see it for alpha-tocopherol. It was really specific. And it wasn't as if, the concentrations were that different among the carotenoids. It was something specific to lutein that we saw this relationship. And again, once again, for the other measures of cognition, we see the same thing, where lutein either approached significance or was significantly related to these cognitive measures. So the bottom line is, does it work? Is it real? I mean, I showed you a lot of cross-sectional relationships. What happens when you actually improve lutein status? And so that's the last study I'll talk about. And in this study, we looked at a double-blinded, placebo-controlled randomized trial that looked in healthy older women. So they were recruited because they were healthy. We really didn't recruit for cognition, which is kind of a problem at our center because people who walk through our doors tend to be really aware, and they want to do nutrition studies and help the world and all that. And so they were pretty with it, these women, because the primary interest of the study was visual function, and we did cognition because of our DHA intervention. So the 4 interventions, we had a placebo, and a placebo match for both lutein and DHA. We had lutein alone at 12 milligrams a day, which is a dietary level. You can easily get it with 2 ounces of cooked spinach, but problem is most people don't get 12 milligrams in their diet, but you can get it easily if you eat a lot of green leafies. We had a DHA intervention of 800 milligrams a day and a combination of the two. And the reason we chose these two was because of their relationships to vision. And so DHA, we did the cognitive measures because they were donated by a company that asked us to do this because of the DHA. So we weren't thinking about cognition because actually I showed you all the other studies. This is the very first study I did, and I did all the other ones to see what was going on. But anyway, so we looked at executive function, and we looked at differences from baseline. And so in this case, a higher number is better. And with each intervention, there was improvement in cognition, which is really something you don't see a lot in the elderly, because if anything, you just don't want things to go down. And to get things to go up is like a big bonus. And so that was nice. And then we looked at learning. We did a test for learning, and it was the combination of lutein and DHA, and in this case, a lower number is better because they learned faster, that they did better. So lutein plus DHA did better, as well as a short-term memory test. So you may ask, why is that? Why together they're working better? And I like this. I like it because we shouldn't think of nutrients in isolation. They are really working together. And when you think about DHA, it's this long molecule with a lot of double bonds, and when you ever have that, it's going to be it's highly oxidizable. So it's really nice that you pair the two together, because I told you lutein's an antioxidant. DHA can also affect lipoprotein profiles, and the only way lutein gets anywhere is on a lipoprotein. That's the only way you find it in circulation, so that's how it's going to get to tissues. And most of it's going to be on HDL, and that's the good one. And so that's going to affect it getting into neural tissue. DHA is also anti-inflammatory, and so that's nice because lutein doesn't like inflammation. Status goes down. So there's a lot of different ways that we can think of why this makes sense, is because what we know, what their roles are as far as the physiology. So in summation, I told you that macular pigment density is related to cognitive function in the elderly. And this makes sense because it's actually a biomarker of what we find in the brain. And that when we look at brain levels, that we found that higher levels were related to better cognitive performance. And that with supplementation, we found an improvement in cognitive function in older adults. So here's a sum of everything I told you, all the different relationships. And the piece that's missing from this chart is what happens when you eat more lutein. Are you going to get more in the brain? And that's a hard study. But the assumption is you do, because of what we know. With an intervention with lutein, with increased lutein in your diet, you do increase macular pigment. And the assumption is that if it's getting to the retina, it's going to get to the brain as well. So that's the end of my story, and these are all the people that helped me tell it. So thank you.